so I'm going to be talking about the so-called ARMS workflow, which is dealing with photographic and genetic data that comes from these autonomous reef monitoring structures, and with an emphasis here on the NIS colonization of the European waters. And so this is the sort of, what is this particular workflow, the science and the data part of this workflow. Right. So I will explain what an ARMS unit is. I'll explain what the ARMS MBON project is. I will tell you what samples and data that we have. I will tell you where the data are archived and how we're linking them together. Then Matthias will pop in and explain the science that we can do with these data. And then I'll come back to say what it is that we would like the workflow to do and why. So an ARMS unit is an autonomous reef monitoring structure. It's essentially, it's a stack of settlement plates. They come essentially more or less off the shelf. They have a standardized construction and a standardized setup, and they are reusable. And this is what one of them, a fresh ARMS unit, looks like. They're placed on the sort of hard underwater surfaces of the sea bottom or a harbour wall, to encourage the surrounding colonizing species to sort of settle on and in between the plates. And we leave them in place for months at a time, shorter periods if we're looking for NIS, longer periods for more general biological monitoring. And this is what an ARMS unit looks like after it's been down there for a while. Once these units are retrieved, the plates and their colonists are photographed. Then the plates are scraped, filtered, and they are preserved, and they're sent off to be sequenced. And these are the three types of sequences that we're dealing with at the moment. And these units were developed under the Census of Life Marine project. So they're not only used by us, they're used by other people within the Global ARMS project. The ARMS MBON project, which is the one that is being run or started off under Istanbul Plus, which is a project which is running under EMBRC. And we're running under one of the joint research activities of Istanbul Plus, which is focused on genomics observatories. And we are a network of about 20 partners, which are placing these ARMS units within the vicinity of marine stations, ports, marinas, and LTER sites distributed over Europe and the polar regions. And the aim of our project is to assess the status of and changes in hard-bottom communities of these near-coast environments using a mix of genetic methods and image analysis and visual inspection methods. And we have this paper here, which you can look at for more detail. Now, the samples that we collect, we have material samples which come from the plates themselves. So as I said, they're scraped, filtered, preserved, and they are sequenced. And there are backup samples which are kept in the labs of the partner institutes. Then we have observation data. So this is spreadsheets of observations which have been made visually, either during the retrieval of the plates or during visual inspection of the plates while they are before they're scraped. And also a visual analysis or a manual analysis, human analysis, let's say, of the plate photographs identifying species on them. Then we also have digital data. These are the photographs that are taken of the plates in the lab, which are then later analyzed either by eyeball analysis or the software. And then we have the meta-barcoding data. These are the raw sequences which are produced by our partner HCMR. So this is this is an image of one of the plates with some identifications done visually. Then we produce also process data, of course. We have process sequences, so the raw sequences are cleaned and clustered to produce OTU tables or ASV files with taxonomic assignments, which we match to the worms catalog. There are also analysis done of the images. At the moment, we've really only got visual analysis of the images, but we've also been testing different softwares for doing a sort of semi-automatic analysis of these images to get species identifications, which will also then be matched to worms, and to which you add some kind of a measure of the quantity of what it is that is on the plate. So we have a map of our partner sites on the right. Up to today, we have one test year of ARMS, that was in 2018, and then we have 2 full years of running, and we're in our 3rd year now. We currently have 21 observatories, and per observatory, there are 2 to 4 sample events that happen per year. So overall, we've got about 130 of these now. Each sampling event produces 3 material samples, a mix of sessile and motile fractions. There are dozens of images taken for each sampling event, mostly of the ARMS plates, but also of any interesting specimens. And of the material samples, we produce 9 FASTQ files. So overall, we have about 1,200 of those already. The data are archived. So we have a data management platform that was used by the project partners called Pluto F, and this is where we deposit our metadata, and we also deposit our photographs and any visual observation spreadsheets. The data are taken down from there and archived in FLIS's Marine Data Archive. This is for long-term archiving and also to make the data publicly accessible. The sequences are stored, the raw sequences are stored in ENA. We're looking at the moment for an appropriate archive to store the ASV sort of OTU data, the cleaned and cluster sequences, because it's important that we also make these publicly available. And we're also looking for an appropriate image archive to store our images, because they can be used not just for the purpose that we are putting them to, which is to identify the species on them, but it's possible that other people would like to use these images for some other serendipitous science. Since the data are spread out all over the place, we link them together. We have a standardized log sheet with various information in them. This log sheet is open access and it's machine interoperable, or will be machine interoperable very soon. And this log sheet can be obtained from the metadata record in the Integrated Marine Information System, IMIS, which I have the link to here. And all the physical data files, so the photographic files, the sequence files, will be made after a short moratorium period. They'll be made open access, and then any species identifications that we will make from these will be published eventually via Eurobis, so within the Darwin Core Archive format. We are still struggling a bit with this because we need to be able to link multiple lines of evidence, so a species that comes from a sequence and an image and a visual observation. We need to link these all together to the data they come from and to the species they give you within the Darwin Core Archive format. And this is something that's currently a little bit of a problem, so it's a work in progress. So this is a screenshot of the top of the metadata record. And this is what the log sheet looks like. So we have, in this log sheet, we've got all the time information, the geographic information, sample IDs, the particular SOPs that we used for each fraction, preservative. We have a list of the runner session codes of the sequences, and we have links to the photographs and any visual observations for each sampling event, for each partner, for each year. Okay, so now Matthias will take over for a bit. I will just scroll forward as you tell me to, Matthias.
There's just 2 arms, and you see that you get like, today we have looked at more, we get between 70 and maybe 100 species per arms. And what is interesting to see here is that a lot of evidence is coming from the images as complementing the sequences. So we think it's a really interesting approach to compare and have both lines of evidence available and be able to integrate them. Of course, the sequence identification abilities depends on the reference libraries, and the image identification ability depends on the taxonomic background and expertise we have in the network. So all these both evidence lines, they can grow in the future depending on how much we expose these data to other scientists. Okay, let's go to the next one. Yeah, so that's 16, and just in these arms we found 16 non-indigenous species. Most of them came from Crete. So we can flip to the next slide, Katrina. Yes. So here you have a list on the left side, the species list, and we can trace it back to the source, to the deployment, all the way, And actually, there was one example out of the 15 non-indigenous species we found in Crete, where we could actually get a match on an image. So there was a signal we got from the sequences, and we could trace it back to an image where we found that little snail, a nudibranch, crawling around. So that was a really nice story. And we have also in Sweden now a similar case where we can make this connection. So let's go to the next one, Katrina. Next one. We can go to the next slide. Yeah, so this is just that we are now starting at the beginning of this year. We start to explore also this on the arms we had on the Swedish west coast from 2020. That's last year. And here we had deployments made in marinas, in ports, and also in protected areas like pristine sites. And you see these sites in this little on the map on the right-hand side, and then you see a table with the number of species that were non-indigenous based on what the species lists that are relevant in the North Sea regions. And you see there are many of the species, we know them. So here it's really a matter of monitoring knownness. But then you have these orange ones. These are in the table, the orange ones. These are the ones that we didn't know exist in the country. So they are newness. They were not. We did not know they were here. And then we also had some of them, which are green ones that they're cryptogenic, so to say. We don't know if they have been here and they haven't been observed for a long time. So you also find some rare species where it's not really clear if they are invasive or not. But in any case, on the left-hand side in this slide, you see that diagram where you see each arms, that is the green dot per place, and you see how many NIS we recover here, and there's always between 5 and 15 NIS we recover. The orange spot is plankton, comparison with plankton samples that we took in some of the places simultaneously, and you see that the plankton usually gives more NIS than the arms, but the arms also give quite a very interesting good number of NIS, and they are not necessarily always the same. So then the arms are capturing, they're working like magnets, attracting species, while in the plankton you're scooping the water column and you're collecting everything that's out there. And many of those species that you pick up in the plankton, you don't know if they ever will settle and survive, but those ones that you pick up on the arms, you know they have recruited, they have been through the recruitment process, and they are about to establish themselves. So let's go to the next slide very quickly here. This is just to start exploring the data set. It's just very preliminary data where we're looking now at the within species diversity in order to understand connectivity along the European coastline. And that's just one example from a species that where we had a lot of sequence variation. We had a lot of sequence diversity, and just in this area we had like I don't remember precisely, but more than 40 ASVs. So these are haplotypes basically, and now you can see and start plotting these haplotypes and see how they connect. For example, in this place on the map called Industrial Port, we pick up some haplotypes that are very unusual for the area. They don't exist anywhere else. So here we may have a signal that is a ship actually transferred haplotypes from another place, because that is the largest industrial port we have on the Swedish west coast. So these are kind of the patterns that we are now able to investigate. And now the question is, for how many species can we do this? And these the two graphs on the left side, you can see that we get a first feeling of this. The left graph shows the frequency distribution of the diversity of ASVs, basically the haplotype diversity for all the genetic terminals we have. So we can't put names on all of them. And the one on the right-hand side is the one where we have species names to it. So we are looking basically at about 400 species where we can do population genetic studies along the European coastlines with the sampling we are starting to build up now. So that's all I want to say. If you go to the next slide, that's the last one, that you see that these different ASVs and how frequent they are, they can be used for two different things. On this circle, this pink circle on the left side, basically our ASVs, they're very rare and a few of them. So this is an indicative for species that just came in from another continent and there's not a lot of diversity there. Or these are species that are by nature very rare. And then on the tail we have in the frequency distribution, these are species where we have a lot of ASV diversity within species, and we could probably be able to trace them for their entire distribution area in Europe and understand how the connectivity works for these.
So this is the science that we can do. So this is the data that we've got. This is the science that can be done. And I have two slides to discuss what it was that we asked the LifeWatch ARMS workflow would be able to do. So since we have this master log sheet, which records, links all the data that we've got together, it says when they were taken, where they were taken, and gives links to where the physical data files themselves are held. The first thing we asked the workflow to do was to access this log sheet. It gets it updated, not frequently, but regularly, and so therefore it has to be accessed fresh each time the workflow is running. And from this master log sheet, the person using the workflow will be able to see what ARMS data is available, and they will be able to choose which ARMS data they want to work on. They can choose a range of dates or locations. They can choose particular raw sequence files they want to work on, or images that they want to work on. And this little recycle symbol that I have here, its purple recycle symbol, means that this is something that we would want people to be able to do several times within any particular instance of the workflow. Then we would want people to be able to choose a particular workflow path. Now, initially we asked for 3 different paths throughout the workflow. One was to take the raw sequences and to clean them and do the taxonomic assignments, so produce the OTU tables and the taxonomic matches using PEMA code. And that's what there is in the workflow at the moment. We would also like the workflow to be able to take the cleaned sequences and process them through some other taxonomic assignment codes, let's call them, such as BOLD or something else. And this is something that is not yet incorporated because the workflow has to start somewhere. And then we wanted to have an image analysis path so that you can do the analysis of the images using a particular software, which we have to yet choose. We've been testing out some software. The ARMS images are a little bit difficult to analyze because they're not quite the same as other images that a lot of community codes are used on. And it's important to be able to do all of these because of the type of science that we can produce needs to be able to work with the sequences and to work with the images and to potentially work with more than one type of species database or taxonomic database, sequence database that you match against. Because we will be sometimes looking for only alien species, for example, or species only from a certain area, or use a different set of libraries to see how good the results are. Then we want, for all of the outputs from the sequence analysis or from image analysis, the taxonomic assignments will be checked against the worms taxonomy, and there will be a check on the alien nature of the species using the RIMS library and the WIMS web surfaces. And then we would like, as a final step, for the outputs from all these species and image analyses to be potentially pushed through a biological statistic analysis using the RV Lab software. And this is something that's not yet incorporated. We still, on our side, we still have to do some work for this. And then finally, it would be nice to include a step where the outputs from this could be formatted into Darwin Core Archive so it could be published on Eurobis. And this is also something that we still need to provide input to the LifeWatch IJI team. And then finally, it would be nice for all the user to be able to download and archive the results either on their own disk or on the LifeWatch catalog.
I'm Antonio Jose Saim, IJI. I'm the LifeWatch ICT Core Operations Coordinator. I've been involved in managing many of the teams that have been working in the different workflows. So let's start with the presentation. First of all, some forewords about all the ICT presentation, not only this. The point is that in each presentation, we are going to explain some technical aspects associated to the specific workflow, but also we are going to explain some elements that are common to all the workflows and are part of the initial Tesseract architecture that we have been testing using this initiative, Alien Specific Initiative, just to create, develop, and test. So let's start with the ARMS workflow. Okay, as Katrina has explained, this is the initial validation case that was defined for this workflow. There are different areas where you can insert files, select file, edit files in the initial idea. We choose some parameters for working with the PEMA code and also with worms and brims. And the objective is not only to have a final output, but also to integrate the RV lab. It's not fully integrated. We are actually testing that, but it's not integrated in this initial version. Well, let's talk about the current implementation. The user is going to be presented with a very simplified image of the workflow here in the center. And the user could insert files to analyze, uploading it, or in the future, we are going to integrate external services to get that files. And also can select a file with all the parameters to run the PEMA code. And on the other side, can edit each of the parameters with a very comprehensive dialog where all the parameters is selected and you can change it. Once these elements are integrated, the system asks for the information needed by the GRIMS interface about the area to identify to work on the system and launch the workflow. Once the workflow is done, you can see that the different elements have been executed correctly with this tick, and also you can explore the results, not only the files, but also the result of each of the execution units. Also, you can download the different files, the results, and the log of the execution. So you can analyze what is the situation of there is any warning in the services that are being executed. All in all, all the workflows is built using what we call wrappers. A wrapper is a way to integrate any potential executable call or external service into the workflow engine. So we have two aspects. The first is how to integrate, parameterize the wrapper to follow the workflow. And on the other side, we have a specific execution environment that is dependent of the implementation and can be integrated in our system. This kind of execution could be a local executable or a Docker image or external services. More on that later. All in all, our first goal, not only in this workflow, but in all the workflow, was to generalize them as much as possible. Because generalizing those wrappers, we can reuse them in different workflows. Here we see each of the wrappers we have developed. We have parameterized them with some metadata. We have identification of the type of file it consumes, how it's executed, in this kind the technology, that is in this kind R and Python, if this wrapper has a graphical user interface integration, and also an initial evaluation of the technology readiness level of this concrete element. This is a standard way to analyze systems. In this case, for this, the level is 5. It's intermediate for a demonstration. We have also developed the PEMA sequence retriever that consume the ARMS files and the parameters and produce FASTQ files. This is also a local executable. This one is one of the initial Docker images because the PEMA runner is really, really complex. It's a very big environment with a lot of dependencies. So we encapsulate all the dependencies using a Docker environment. And through this Docker, we execute this runner. Also, we have developed ARMS file extractor, the task check, executing the work services, the invasive check using the green services. We generate the output, and we are now working on how to run the RV lab, because also the RV lab is really, really complex, and we need to encapsulate it into a Docker environment. So that's the idea with the different wrappers that we have deployed to create this workflow. But there is something else. Something else is about that also, if you want to run something like this, so complex, and in the context of, internet is not about the World Wide Web, it's about the Wild West right now. And you are aware of lots of attacks last week, the previous week, in different governmental and private infrastructure at large scale. So from the very beginning, we need to maintain and analyze and deploy specific elements in the architecture to assure that there is no problem with tampering or just modifying services or modifying files, on corrupting anything, or just taking it down. So this is a very conceptual view of what we have deployed. From the point of view of the user, you are here with your browser. Okay, perfect. And you can think, okay, I need this file from this site. I need to execute this element from this other service. And then I'm going to combine them. I want to share them. Okay. As you know, you cannot do that in a secure way. You need to provide all those elements in a controlled environment. So what we first deployed was a very strongly secure environment where the identification of the user, the authorization of running each workflow, each environment, sharing files, etc., 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 is fully managed. And we continuously monitor the access, modifications, every single aspect. In this way, we can block and prevent malicious access to the system. There is only one element that we have A partial restrict access is not through this first advanced perimeter security, is to the second perimeter security area, where is running the different elements. Which elements are running here? The elements that are running here are first, the workflow engine is a very complex engine, very extendable and with scale-out capabilities, very flexible. And in another presentation, it's going to be presented. Also, we have a distributed storage service, very robust, and that can hold a lot of information in a secure way. This is the only element that right now we have provide a partial access in a very restricted way. On the other side, we have three kinds of wrappers or executables. Native execution services that is running side by side to the workflow engine. There are small elements and small applets or running code that you know, it's better to run near the workflow engine because it's effective and it's not complicated to maintain that. We also provide an internal distributed execution service running the Dockers. Sorry, those Dockers are internally distributed, are completely secure. You cannot access them outside this secure channel, but allows you to execute complex, very complex and very big environment and services. Finally, we have an initial remote service invocation. This initial deployment is running cloud services and external services through a secure channel. This initial deployment is associated to different users, but in next releases, those external services are going to run through service level agreements with LiveWatch Eric, with more capabilities and a specific prioritization for the users. Finally, of course, all the monitoring services to maintain and assure that this environment is also perimeter security. Finally, we are now starting to work in a way to access, to deploy high performance computing jobs through these workflows. Of course, the user never is going to access those high performance computing environments. Never. Those elements are really complex, have a specific command lines, and also people need a specific permission just to access, manage them. So we are now working in a gateway, a specific gateway that on one side is like a cloud service, an external remote service invocation, as well, other services we are now integrating, but on the other side, specific elements in the gateway transform those services into running jobs through a specific SLA that we have seen with different providers. For example, Picasso Supercomputing Center here where we are now. Well, I think that that's all.
My talk is regarding the verification process for research infrastructures, and in particular the case of the LiveWatch Greece research infrastructure. The Hellenic Center for Marine Research is participating in the MRefer project, through its national LifeGoals Greece research infrastructure. And at this point, I would like to thank my colleagues Eva Hadjinicolau, who is the scientific responsible for the EMRIFAIR project, and our IT manager, Emanuela Panderi. In the Hellenic Center for Marine Research, I work as data manager, and in recent years, I often come across with the concepts of open and fair data. As manager of scientific data, it's really difficult to deal with these concepts because although researchers acknowledge the benefits of open data, data sharing practices are still limited. It's worth mentioning that the global online survey in 2017 of more than 1,000 researchers found that less than 15% of researchers share data in a data repository, and more than 80% share their data directly to their collaborators. That means that they are using methods such as emailing data files or cloud services such as Dropbox or Google Drive. Open and fair data are not identical concepts, but both make clear the importance for sharing data and saving cost and time between scientists and users. Speaking about fair data principles means that data should be findable, accessible, interoperable, and reusable to the greatest extent possible. These principles apply not only to data, but also to metadata and supporting infrastructures. That's the reason why trustworthy data repositories capable of curating fair data for scientists play a crucial role in the evolution of open science. It's true that it's difficult to define fairness, but of course there are steps to be followed in order to improve the fair status of existing repositories. So it's important to pay attention to good practices for such repositories. And here I have some examples that repositories should support standardized metadata formats, should have a search web page, links between data stored in the system should be supported, usage license associated with the data, and support publishing open, closed, and embargoed data. Repositories should preserve information about data provenance, which could be stored in metadata. And last but not least, humans and computers should be able to exchange and interpret each other's data through commonly used control vocabularies and ontologies. LifeGoals Greece research infrastructure, and in particular the Mediterranean node of Ocean Biodiversity Information System, MEDOBIS, provide reliable and quality control marine data sets from all over the Mediterranean Sea. MEDOBIS hosts a plethora of marine data sets, both data and metadata, and already most of the FAIR principles are applied. Regarding metadata, LifeGoals Greece research infrastructure gives emphasis to metadata availability, even if the data are not available. A metadata document with mandatory fields such as dataset title, citation, persons involved, etc., as well as suggested fields, is created and distributed to all marine data providers prior to the submission of their data. Also, it's important to highlight that metadata are always open to public. That means under CC0 license. Regarding data, LifeGoals Greece publishes its data through a free open source software tool. And in order to publish datasets, data records are standardized in Darwin Archive. WORMS taxonomic mapping is used for location the marine region applications, and for environmental information, the methodology. Also, detailed provenance, citation, and usage license is always available. To conclude, LifeGoals Greece research infrastructure is findable as it is a digital repository and accessible through its web page and portal. Its interoperability is guaranteed through commonly used control vocabularies and methodologies, and its reusability through its data policy and sharing agreement. But future challenges do exist in respect to FAIR principles, such as common strategy and procedures, especially methodologies used, and certification process in order to enter to the European network of trustworthy repositories enabling FAIR data. It's really very optimistic that Lagos Greece research infrastructure has already invested in fairness of data. Undoubtedly, there are more steps to be done, and we are engaged to fulfill them through our participation to the EMBRI-FAIR project. In addition, more effort shall be given to define a good semantic model that could represent a consensus view in a particular domain, and in our case, for the marine domain. It is proposed to define a common set of indicators for all lifeguards, common facilities and centers in order to assess our fairness status, and also to define a common certification framework for our repositories and services. Thank you very much for your attention.
But we get one question here from Kemal Khan Bizel. Is there any available data on some pressures or stressors affecting on the sampled species and the sampling sites? And I think that's a question for Katrina to start answering. So where are the stressors data?
In your head? I know that certainly. So we're in ARMS, we're not collecting associated data or environmental data for the reason that these ARMS units are down there for a long time, and it's in principle possible to collect all sorts of other data, but in practice it's extremely difficult to figure out how to do it and how to manage it. Although there are one thing that is sort of on our plate is to look at monitoring observatories that are located near our sites to gather data, to harvest data from them that we could perhaps use when we start analyzing. This is something to do in the analysis phase rather than in the data gathering phase. And as for the, well, and then Matthias can also add to this question, or to this answer, I should say.
Yes, yes, okay, I can come in here. That's because it's when you're building up these pipelines, it's not the point that they should do everything for everybody. It's a point to have those pieces of analysis that are especially difficult in terms of a lot of computation, a lot of complexity, and also in terms of data standards that you want then to enforce to be able to make the data interoperable and accessible to everybody. This is where you want to channel them through this piece of infrastructure. So in principle, the output that AJ presented, you can combine them with the input that Katrina presented. That's the metadata on the place, and then you can add all kinds of information yourself, like what kind of temperature was there, what kind of environmental condition, how strong a pressure A or pressure B was there. For example, we have arms, they are under extreme pressure on in harbors. They are just in the middle of the industrial harbor place. There's a strong pressure there. You can add any pressure you want, and that is up to the individual scientists to do it, while the infrastructure is just helping you to analyze the data in a standardized way to allow to compare sample X with sample Y, and that is the magic that can appear here when you enable this.
I just
AJ,
want to
add, oh yes, Katrina.
Yeah, so this is one of the reasons why we have a data manager in this project. I'm not a scientist. And that is that in order for, there are dozens and dozens of types of analyses that you can do with these data, but what we want to do is enable other scientists to be able to do their analysis without having to spend half their time just finding the data, figuring out what format it's in, converting it to the format they like, understanding what this column means. And so we're spending a lot of time making sure that these data are semantically annotated. So every term that we add to our data set is explained. machine interoperable, so you can write your own R code or Python code that can access this data, and it can access every year's will be exactly the same. So we want to make it as easy as possible for you to be able to, you, the scientists out there, to be able to access these data in an easy and understandable way that will never change, so that you can do these other analyses yourself, and you can do them easily without having to spend. So all your thinking, all your brain power will be on the science, not on understanding the data.
Yes. AJ, do you want to add anything on this from your side?
Oh, yeah. Our final goals is to provide the most complete capabilities to our users, to scientists, not only standardized workflows that are really, really needed just to check, recheck, review, and replay different analyses in a certified way, you know, but also to provide tools to make them capable just to analyze as they want. And so one of the elements that we are going to integrate soon are specific RStudio and Jupyter Notebook environment connected to the files, the input and output files in some specific steps associated to the workflows. It's a work in progress. Now we have analyzed some different alternatives, but we want to provide them all those capabilities. Okay, the point is, if you follow all the steps in the workflow, we can provide the provenance. If there is some steps where you are going to work and play with your files, algorithms, modules, etc., 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 we cannot assure the provenance. But if you agree, once you have finalized all this analysis, to upload those analyses, those modules, those intermediate files, we can get some provenance. So that's the point. But our objective is to provide all those tools as complete as possible. And of course, it's not easy because there are a lot of different tools, different scientists, but more of them are working with RStudio or Jupyter Notebooks, but also some of them are working with some specific codes in C++, etc. So we need to provide a flexible environment for that. But we need some good practices associated to fairness, etc., but also to the provenance. That's
yeah. Thank you, AJ. Just to expand on this and coming back to Kemal's question, because he had a follow-up question, it can be an absence-presence categorical data type that would allow you to make these kind of analysis once you have finished the primary genetic cleaning up analysis and the taxonomic assignments. And that is true, but and as Antonio said, that when the RStudio, the RV lab will be attached then in a later stage, you can upload these categorical data yourself. And if there is a need for it and sufficient interest, you can also connect that to a database that contains these categorical data. That's a question of the users who have a strong case and say, like, we want this, because it's not only one, it's many of us who want to do the same thing. And then I'm sure that it would be could be implemented. Okay, so I have some more questions. So since we already started asking Katrina so many questions, so I go to AJ first here, and I'm just wondering about the workflow structure you have built up on in the ICT side. How compartmentalized is it? I'm just thinking as an example now. This is very much customized now towards the analysis of non-endogenous species. So you have, after the taxonomic assignments, you have an automatic follow-up checking against RIMS on all the invasive species. But let's say there's now a researcher coming who doesn't want it, doesn't care about the invasive, wants to analyze all the biodiversity that you find in the samples. So he doesn't want this method. How easy it is to
There are two different elements here. There are two different elements here. First, the engine and the environment is capable to evolve and to add many of those elements. Some of them are now in testing, checking, and the like. But we needed just to start deploying an initial workflow, an initial instance, a first version, and we want the feedback from the users, which are new services that they want us to integrate, check them, validate them, and then we deploy. And also that's one of the reasons because we want to deploy an initial sandbox where scientists can start working with all those elements, test all those modules and libraries, and say, okay, these and these and these elements are good enough for us. Please validate them so we can automate them into a workflow with all the quality assurance, etc., etc., etc. So we are really open to all those feedback. We want them, but we wanted to start with some initial step and say, okay, start working with that, give us the feedback, and we start evolving any of them. The environment is really flexible, very powerful, and also we are indeed we are now migrating some of those specific steps in a standard way to Dockers running in different instances, so we can not only be flexible with the capabilities of each of the services, but also with the escalation of those services, because we have some initial capabilities, but once people start working with that, we need to scale out in different environments. So we have analyzed the way to deploy all those services using different providers from live sharing, but also external providers in a secure way, because that's the initial point. So we are open to all those elements, really, and we want as much as possible all the feedback from the users and start working and evolving all those elements.
Thanks. Yes, that's very good. Okay, so we got a new question coming. Anyway, what about reuse of individual workflow components and chaining with external services and components?
Yeah, that's the reason our unit is the wrapper. The wrapper, we have the capabilities to reuse that wrappers. So an initially thought was to standardize some of those steps as reusable wrappers. And the second step that we are now working on is to standardize the way to automatically deploy those components and deploy a composition tool. We are now testing the initial capabilities of a composition tool where user can select different wrappers and combine them in its own workflows. So step by step, is in our roadmap to deploy a composition tool for personal workflows.
Okay, I think that is probably answering the question. It is also where my original question came from, like how much you have compartmentalized workflow so they can be changed and readapted to, because that is the flexibility that we probably need. But there's a practical question associated to this. So maybe Antonio, you're not the right person to ask, but let's see that now you have had these scientific use cases and the technical development in very tight, and now you seem to be entering the next stage where you want to expose it to the wider scientific community and collect that feedback. And what is the format you're choosing for that? I'm just wondering, this connection has to be like, sometimes it's individual scientists, sometimes it's more research groups. Do you want to have that communication and that contact through conferences, workshops, like small proposals, hackathons? And also, do you think you need some training? I mean, that like all these scientists, they need to have some background and training. So how do you see that, and what's the time scale? Do you want to, will you enter that interaction now with the wider community, and how do you see that interaction with the scientific community?
Okay. A year ago, more than a year ago, we defined an initial way of working. That initial way of working was there is a tight group of scientists focused on analyzing those kind of workflows and how to deploy them. A tight group of developers defining the best solutions possible in that moment, and we held different workshops where we work together and start defining not only workshop but also working groups defining how to improve and refine those workflows. Right now, once we start spreading the access to these workflows, we have deployed some specific connection tools, okay? But that's not the question. The point is that you need, from the scientist community, we need some leaders that say, okay, I can gather these aspects, so we can manage the communication with that leader or groups. Okay, we need that. And also, scientific community is wide, very diverse, and they can manage those groups as they like. But we need some contact points in these communities, just to make sure that the requests, the requirements, and the capabilities are the most common for all of them in the way of working. Okay, and so we have the people working on the technical aspect associated to that, but we need this kind of connection. We have an initial group with different leaders, but if we start now to spread all those elements, we need a way to integrate more leaders, different working groups in different requirements in the scientific workflows. Perfect, but we need that. That's the real problem. It's not a problem, it's the issue that we need to have resolved by the scientific community. So workshops, training, of course, all those elements are needed, of course, but we need leaders for each group as they want to organize, but a contact point for each of those groups.
Good. We have to make some advertisements and
we have a question from Christos. I'm gonna give him the floor, okay?
Yes.
Hi, can you hear me?
Yes.
Yeah.
Great. It's not a question. It's actually a clarification.
I don't know. It's a little bit. I have coming back in my
Yes, yeah.
in my sound anyway. This will be the case in our next 5-year implementation plan.
Okay, that answers
exactly the way that AJ was describing.
Yes, that's very good, and it's a very important step, I think, to in the next step. Okay, well, thank you very much, Christos. That sounded a little bit like echoing. It sounded like the voice from above that answered everything in one sentence. So I'm very happy about this. Okay. Unless there's any other questions now or anyone else who raised a hand, I can go to my next question to both of you, and probably also to Dimitra now. This is about FAIR, basically reflecting a little bit on the presentation that Dimitra gave. And I'm just wondering now, Katrina, starting with you first, if we see these workflows and this infrastructure as a verification engine, then my question would be like, how do you, on your side, in the data management, how do you see that this infrastructure can help to make data more fair, like the biological data? Like, where do you see the critical points where you can make a difference?
Well, I see. So this particular, as it is right now, the workflow starts from data that that is published, and then does analysis or allows analysis to be done on those data, and then those analyzed data are are published. So every step of that process needs to be fair, but the data gathering part of it, which is what we're in control of, is done separately to this workflow. Now, since the data gathering part of it is being controlled by us, we are able to determine that it's done in a fair way. And in fact, by having to make our data accessible to the workflow, we are having to add machine-to-machine elements to our data, for example, semantic annotation of our data files, which makes them even more fair. And then the fact that the workflow will be able to trace the full provenance of the data that are processed within the workflow is extremely good, because this is something that's not really done by scientists working sort of individually. For various reasons. In order for a project such as this one, because there's many different types of data that we are producing, and a lot of it, managing it is extremely important. If you manage it properly, then you can add all the fair elements to it. And the best way to manage it is in some kind of a centralized workflow. It would be very nice. At the moment, we're still playing around with getting the data out of our data management system and then organizing it so it's machine interoperable and fair. And it would be nice, in fact, if we had a workflow that would do that for us. If there was an IJI workflow that was a data management workflow that would allow us to plug and play with, for example, putting in Pluto F and then adding in our commands for reorganizing the data. So it would be very nice if there was, if LifeWatch could have a data management workflow as well. Could then that would be an extremely useful thing for the community, I have to say, because then they wouldn't need data managers like us to do it for them. Anybody could then do it, and it would be an awful lot easier, because it's taking you a long time to get to this point. So that's my request to LifeWatch.
Yes, a point. You know that there is a data management plan working group, okay? And this data management plan working group is really is working on defining some kind of machine actionable data management plan. Not only a list of books and recommendations, but machine actionable elements. That's one of the goals. And also is associated with the LifeBlock infrastructure we have been deploying that provide the provenance and all the trace of the transformation and access and the like. Is very connected to that.
Good. We got a question from Wiebe coming in here. He refers to the NCBI gene bank services that are very simple requests, and they can be answered in a standard and user-friendly way. And he wonders if LifeWatch is going the same way. Maybe Wiebe, you can reiterate here.
Can you hear me?
Yes, I hear you. Yeah.
Okay. So the point here is that all what I have heard seems to reflect fairly complex data streams for which also the users have to be professionals to make use of all these services. Now, when you look, for instance, at NCBI GenBank, or you take data sets like OBIS and GBIF, you can go to those websites and you ask relatively simple questions. I want to know the distribution of this species, and I want to know it from the 1980s onwards, and you can specify in a questionnaire where and what you want, and plop, there is your answer. In GenBank, it is organized in the same user-friendly way. Of course, there are the sophisticated services behind it, but lots of questions are relatively standard and simple questions that can be answered also in very simple, standard, user-friendly ways. So is LifeWatch foreseeing that? Because that is, I think, a major user thing. And it will also become then available for citizen scientists and people not professionally busy, but wanting their answers as well. Thank you.
Yeah, of course. We want to deploy a specific catalog of services and components and how to integrate them, or just ask a specific question. Of course, we have seen and we are aware that we need to provide a broader spectrum of capabilities, from the more complex one to very simple one, microservices, micro capabilities. But both of them, we want to deploy them, and we're aware of that.
Thanks.
If I may add a point here, it's that I fully agree, and that's the crucial role of repositories. So users and scientists can be assisted by specialists. They have the knowledge, data manager are there and can support the scientists to open and verifying their data.
Yes, also to finally add to Weebe's question from my side, that there is a lot of interoperability with existing architectures like NCBI and GBIF. So all the data that come out in terms of FASTQ files, FASTA files, they will end up into genetic repositories. Those ones that create species names will go into OBIS and then eventually to GBIF, where you have these interfaces where you can ask these very simple questions. So we also have to be careful we're not reproducing something that already exists, rather than linking properly our data infrastructures.
Yes, I understand that, but there might be niches that are not yet covered.
Exactly. That's something that when the user's making a strong case, then I think there's a good chance that this will be developed in LifeWatch then. Okay, that was about the technical side of fair data. Now I'm wondering about the cultural side of fair data. The problem that Dimitra described, that many scientists, they don't see the benefit, nor they adopt the culture of sharing their data, because they don't see that they don't get sufficient credit for it. They see more benefit of it keeping them. So how is this arms network, and how is the technical infrastructure of the workflows contributing to capturing data, basically convincing scientists or helping them to share the data openly rather than keeping them for themselves? AJ, you want to start with this one?
From I think that from a technical point of view, the first element is about the kind of license you are going to use to deploy those data. So you need to associate those data sets to a specific author, and also to provide the capabilities to deploy them. So you need some kind of CC1, at least CC1, not CC0, but CC1 license, at least for the developers and the scientists that provide those data. That's first point. The other point is that the provenance infrastructure we are deploying and evolving trace all the access, not only from the provider of the data, but also all the transformations of that of those data through different stages. So in the end, you have a complete trace of the origin of the data, the reuse of the data, the reuse of the results, all those elements. And so once you want to publish them in the final stages, you have all the trace. So from the point of view of the how to really identify who is providing the data and who is responsible for the high quality of the data, from the point of view of the infrastructure, it's not really complex, not at all. But the point is, we first need to agree to use specific licenses, not a public domain license, but associated authorship to those data sets and the transformations. And it's just policy. It's not a technical problem. It's a policy problem.
Yes, I agree on this one. Katarina, you want to add anything on the on this one?
Yeah, in my perhaps somewhat limited experience, I do find a willingness, let's say a personal willingness on behalf of the people to share their data. They understand why it's done, but it's not easy. It's not easy. I mean, it's easy enough to sort of, if you have an image or a CSV file, and just sort of dump it into an archive, but then you've got to record for it. Then you have to have it in a kind of a standardized format, and you've got to explain what the columns are. Then you've got to use, you've got to use the BODC vocabulary to say where your parameters come from, and it sort of and. So now, of course, you have to change the culture from the university level onwards, and this is something within these FAIR projects that we can tackle. We're not talking about a 2 or 3 year timeline. We're talking about a 10-year timeline here, right? But I really think that the only way that we will get a mass take-up of scientists making their data fair, not just open, but actually fair, is if they can do it easily without having to have to spend 2 days a week figuring out how to do it. And for that, they need to have tools which are affordable, so free, in other words, not commercial, or at least cheap, that are affordable, that allow them to do this data management from the very beginning of their work in a way that doesn't sort of add that much of a burden. And this is the one thing that I think something like the EU and these European FAIR level projects could offer. They don't really at this point in time, but I think that really would be something that would make this mass take up, because once data are out there and they're fair, so that they are understandable, they have provenance, then for professional data managers like us to come along and then say, okay, so now we'll take this and we will make it accessible and findable, it's very easy for us. But if we have to take these data sets and we don't understand half of what's in there because it's not been properly explained, and then have to interact with the scientists, then nobody wants to do that and everybody loses. So what we really need is for the EU to pay for projects to produce tools that allow for fair data management from the very beginning.
Yes, I think that was a very good point. Okay, and so I think I've asked all the questions I had. If there's no one else, I have one last question about documentation. So these workflows, sometimes you have to teach the users how they run, like technically, but also what's in there. So what scientists don't like is the black box thing: data in, data out on the other side, and I don't know what's happening inside. So how do you think? I mean, it's not your responsibility, because whatever you wrap up, that's a scientific product often. So but I'm just wondering, in those pieces that you have now, how well is the documentation done? Is there any work that's left to be done? For example, PEMA, how well is it documented?
Okay, the point is we have two different points here. Those elements that we have developed from the very beginning, from scratch, I think are really well documented, really well. And of course, following as much as possible all the feedback with the initial, not only developer, but also people, the scientists just thinking about this and analyzing and generating this initial step of the initial version of those codes. For that, we maintain a specific repository with all the control version, etc., etc., etc. But we know that we need to do more work in the way to document that. That's one of the reasons we have also started to standardize the way to describe those elements as a service, the parameters, and also how it's working inside. So we can not only define and describe the transformations, but also in a way that the machines and all the people can just analyze in a standard way. So we are working with the semantics group about how to standardize the description and also the capabilities of those services. In the case of external services, that's really complex, because we can somehow document some subset of the use cases or the tools or the capabilities of the tools we are using, but it's not dependent only on us. We can do more effort, try to not only to document that, but also to push those data documentation or those changes to the upstream to the original creator groups. But we need to start a closer communication with those groups. That's the point in this moment. Our goal to do that as more comprehensive as possible.
Thank you very much.